All right, if you're deep into your GCSE biology revision, you know that the required practicals are a huge deal. Well, today we're cracking the code on Practical 5. We're going to get right into the science that makes this whole thing work.
So let's kick things off with a pretty mind-bending question. How can something as simple as a small change in acidity completely shut down a process as powerful as digestion? It sounds wild, right? But figuring that out is exactly what this practical is all about.
So here's our game plan. First, we'll get to grips with enzymes. Then, we'll look at the specific challenge with amylase and pH. After that, we'll walk through the practical itself step by step. Then we'll figure out what to do with the data. And finally, we'll look at how to smash those exam questions.
Okay, before we even think about touching a test tube, we have to understand the why. The entire experiment depends on these amazing little things in our bodies called enzymes.
So, what on earth is an enzyme? Well, think of it as a tiny biological machine. It's a catalyst, which means its whole job is to make chemical reactions happen way, way faster. And the most important part of this little machine is a very specific spot called the active site. And this brings us to a really cool idea called the lock and key theory. It's the perfect way to picture how this works. The enzyme's active site is like a lock, and the molecule it works on, the substrate, is the key. Simple, right? Only one specific key can open that lock. This means that for an enzyme, its shape is absolutely everything. If that active site, our lock, gets bent out of shape, the key just won't fit anymore. And when that happens, we say the enzyme is denatured. A denatured enzyme is basically a broken one, and the reaction just stops dead.
Okay, so now that we've got the why sorted, let's focus on the what. What specific enzyme are we using, and what are we going to be testing? Our star player for this practical is an enzyme called amylase. It's a carbohydrase, which is just a fancy way of saying its job is to break down carbs. Its mission? To chop up massive starch molecules into little, simple sugars. Fun fact, you've got it in your saliva right now.
So here it is, the official aim of Required Practical 5. We are going to investigate how pH, so how acidic or alkaline it is, changes the speed or the rate at which amylase gets its job done breaking down starch.
All right, we've covered the why and the what. Time for the how. Let's break down the method. Don't worry, it might look like a lot of steps, but it's actually really logical when you see it laid out.
First up, the prep work. You get a spotting tile and put a single drop of iodine into each little well. Then, and this part is super important, you use a water bath. Why? To keep the temperature exactly the same. We only want to test pH, not temperature. Lastly, you'll mix your amylase with a buffer solution, which is what lets us control the pH.
And go! The second you add the starch to the amylase, you hit that stopwatch. Now, every 30 seconds, you'll use a pipette to take a tiny sample from your test tube and drop it into one of the iodine wells. You keep doing this until the iodine stops changing color.
So, why are we using iodine? Well, it's our starch detector. If starch is around, the iodine turns from its normal browny orange color to a deep blue-black. The moment all the starch has been broken down, there's nothing left to react, so the iodine just stays browny orange. That's our finish line. That's the time you write down.
But, you know, in science, one test is never enough. To really see how pH affects things, you have to repeat the whole entire process again and again, but with different pH buffers each time. So you might test pH 2, then pH 5, pH 7, and so on.
So the hard work's done. You've got your timings for all the different pH levels. Now what? Well, this is the so what part, turning those raw numbers into an actual meaningful conclusion. Your results table will probably look a bit like this. You've got your pH values lined up next to the time it took for the starch to disappear. And you can probably already spot the pattern, right? The reaction was fastest, the time was shortest, right around pH 7.
Now, in your analysis, it's much better to talk about the rate of the reaction, not just the time. Think about it. A really fast reaction takes a short amount of time, so they're opposites. The calculation is super easy. The rate is just 1 divided by the time. This way, a short time gives you a big number for the rate, which makes more sense on a graph.
And this is where drawing a graph makes everything click into place. When you plot the rate of reaction against the pH as a bar chart, bam! The pattern is crystal clear. You can instantly see which pH is the winner. It's the one with the tallest bar.
That graph leads us straight to our big conclusion. Enzymes have a favorite place to work, an optimum pH, where they are fastest and most effective. For our enzyme amylase,
That sweet spot is around neutral pH 7. If you go too far away from that, either too acidic or too alkaline, the active site starts to change shape. The enzyme denatures, and the reaction rate just plummets.
Okay, understanding the practical is one thing, but now let's focus on how to turn that knowledge into cold, hard marks on your exam paper.
You can pretty much guarantee you'll get a question on variables. Just remember it like this. The independent variable is the one I change, in this case the pH. The dependent variable is the one you measure. That's the time it takes. And the control variables are all the things you have to keep the same to make it a fair test, like the temperature and the concentrations.
Another classic exam question is about evaluating the experiment. Because let's be honest, no practical is ever perfect. It can be tricky to judge exactly when the color changes, right? That's a source of error. To get top marks, you need to spot these little issues and suggest sensible ways to improve them.
So if you take away one single thing from this entire practical, let it be this. It all comes back to the lock and key. A pH that's too high or too low breaks the lock. It changes the shape of the active site. And if the lock is broken, the key just doesn't fit, and the reaction is over.
And I'll leave you with a little challenge, a question to chew on. We know pH and temperature are huge factors, but what else could be slowing this reaction down? What would happen if you had the perfect pH, but you didn't have enough enzyme to start with? Thinking about questions like that is how you go from just knowing the steps to really, truly understanding the science.